It's my pleasure to welcome everyone to the next of the monthly ReproIn webinar series. Again, the reminder that every first Friday at 2 p.m. Eastern, ReproIn is running this kind of webinar. Next January 3rd at 2 p.m., just after you're overcome your New Year's malaise, you'll be ready to hear about some new cool ReproIn tool. And if the scheduling gods are willing, we may hear about the tool called Test Kraken next January 3rd, but stay tuned to your email and your Twitter and however else you get news about ReproIn. So without further ado, it's my pleasure to introduce Yaroslav or Yarek or Hey You Halchenko from Dartmouth to talk to us about these pesky things called DICOM data and how to get control from the beginning. Take it away.
Hello, everybody. As Dave introduced, I am Yarik, and if you want to discover more about projects I'm involved or some kind of philosophy, please visit centerforopenneuroscience.org, which is at Psychology and Brain Sciences Department of Dartmouth College. To discover more about projects on the ReproIn agenda, visit reproin.org, and this is a joint effort between many institutions, and it was supported by NIH. Hopefully that funding will be also Repro funding. And before I actually introduce you to the tools, I want first to acknowledge people because many slides here are borrowed from Matteo's talk at OHBM 2018, I think, and also from Michael Hanke, with whom we collaborate on many projects. And of course, nothing could have been possible without contribution from many other people and other lead developers on the HeuDiConv project, Satra and Matthias, and other contributors. You could see from the right what GitHub says, that there is 31 direct contributors to HeuDiConv, which is like a code bed for ReproIn as well. But also, if we look deeper into all the projects which we use within HeuDiConv, it's over 20,000 people, which is amazing. And I want to acknowledge people from Dartmouth Brain Imaging Center, led by James Haxby, because they allowed us to do this crazy thing as to introduce ReproIn the center, center-wide a few years back. So hopefully that would be an example on how you could improve your workflow at your center. So there is a problem that to reach reproducibility, we need to do our computation really efficiently, but we are so busy. And to help guide you through the solution to this problem, it cannot be just one thing. It's pretty much approach which consists of many steps where you could improve piecemeal. And part of our ReproIn training and also other materials are these 5 steps on how to make your imaging research more reproducible. And today we'll touch upon only a small portion of it, which is data collection, where we'll talk about ReproIn and BIDS and DataLat, but also it shouldn't go unnoticed without open brain consent. So we will not cover it, but first thing you need to do is to look at the open brain consent if your consent forms for participants are not including provisions for public data sharing. I would strongly advise you to look at the open brain consent project and maybe adopt one of the existing there examples or the ultimate consent form wording within your consent forms. So let's talk about ReproIn. And once upon a time, there was a problem. Well, we were still busy and data sets were coming in a variety of layouts. So we had no time to understand data of others. And by others, I mean not only other labs, it could have been others within the same lab. So data would have been collected by one student, and then nobody else in the lab could understand without going through the data set and reading some documentation, what is what, and which file pertains to which subject and task, and how to work with the data. So BIDS project, which is Brain Imaging Data Structure, was bootstrapped by OpenNeuro and INCF, and later on expanded into truly community-driven project where instead of being governed by some specific organization or a lab, people from all over the world decided finally and agreed on how to name files, which is quite exciting. And you could discover more by looking at the original white paper, I would say, about BIDS, which was published a few years back, already almost 5. And at the bottom of the screen, you could see an example of BIDS dataset, or part of it, which pretty much summarizes how files are named and what BIDS is about, that it's about having nifty files with neuroimaging data and possibly some associated metadata files shipped along with them. And all the names of the files are clearly prescribed by BIDS specification. What it allows to do is, first of all, to really ease our understanding of data sets which we see. So you've seen one BIDS dataset, you pretty much have seen them all. What I meant by that is that they are all looking the same. So you don't need to spend hours just trying to figure out where to find specific subject or which modality or what protocols were used. And in addition to that, BIDS is not only human readable, but machine friendly. So toolkits such as PyBids, BidsMATLAB could be used to script your use of BIDS datasets. And additional advantage is that you could even check that, oh, this dataset is valid BIDS dataset by using BIDS validator. But in addition, BIDS apps provide you turnkey solutions for BIDS datasets, and that's what lured many people in, because you could use fMRI Prep to do full-scale pre-processing of dataset, really flexible, really versatile, or you could run MRI QC and inspect your data regarding different artifacts and maybe some defects in the data. And that makes it really powerful. You don't need to spend hours just to prepare your data for analysis. Pretty much, if you have a BIDS data set, you could just give it to those tools and have them working without much of effort. But such solution brought another problem, right? We still stayed busy, so we saved some time on not worrying about how to name our files, but we had to name those files. And those who collected datasets before, they had to somehow rename the files so they fulfill BIDS specification. And we needed a solution, right? So we needed a solution which takes a bunch of DICOM files, which is what we get from the scanner typically, and prepare a BIDS dataset out of it. And of course, any solution should be automated because that makes it more efficient. But we know that sometimes in theory, it sounds like a good idea. Oh, I'll just work on automating it for our specific problem. And then automation kicks in and then everything is automated and great. But in reality, when you start any automation or software project, it becomes a little bit tricky and it kind of goes into ongoing development and there is no time left for original tasks. So we decided to automate it and automate it in such a way that we would minimize manual entry. So that was one of the problems with anything where humans need to enter something on anywhere. They would make mistakes more probable than computers or scanner. And then we wanted to organize all the converted data sets into some logical hierarchy, let's say for the entire center or lab. And we wanted through this automation also to extract as much of information as possible from DICOMs instead of manual entry again. And also make it flexible enough so we could accommodate any type of neuroimaging study. And to kind of avoid this problem of automation project becoming really involved and taking too much of our time, we decided to adhere to this principle of open by design, which is join and contribute to existing projects instead of reinventing the wheel. And This is generally good advice, I would say, for any endeavor. So instead of doing a new project, look around and see what other projects exist to solve maybe your problem, not in full, but at least partially. Then you could join and learn possibly better coding and testing practices. And then you relieve yourself from these problems which would arise with your new projects, such as establishing development, release, and deployment infrastructure. If you're joining existing projects, those are probably in place already. And also you could hopefully offload and share user support with bigger community. You might get credit faster and also find new friends and collaborations, or just passion for your evening endeavors when you have nothing else to do. So under this premise, we decided that ReproIn project, which will take care about this automated conversion, will not be just standalone thing, but we will use existing then already solutions such as HeuDiConv, which is heuristic DICOM conversion, where you could create your heuristics, how you convert specific set of DICOMs into whatever layout you wanted. It was conceived by Michael and Satra years back before BITS even existed. And then we wanted to use data-led project to orchestrate management of those data sets. And underneath of HeuDiConv, one of the most important tools out there is actually DCM to NIX, which does actual conversion of a single DICOM series into Nifty file and JSON file with metadata from DICOMs. So instead of, again, reinventing the whole wheel, we decided, oh, we'll just work on top of those tools which already exist in the ecosystem. And this is just an overview of our setup at the Dartmouth Brain Imaging Center, where data from the scanner gets deposited to PEX server, which is running DCM for Qi. But that's, again, that's any PEX server, or you could transfer your data through the USB sticks. It doesn't matter as long as it appears somewhere on a hard drive. In our case, PEX server shares this data, DICOM data, through NFS with the conversion server, where we are running HeuDiConv, which does conversion into bits, and it's powered also by data-led, and all these containers which are used for all the software is wrapped into containers to make execution reproducible and more efficient. And then these data sets, which we convert into bits, get exposed through regular SSH. So you could rsync them or use data-led to better orchestrate your data management across your computing infrastructure, but also share it later on to the web, or to the public archives such as OpenNeuro. So what ReproIn consists of is actually three components. Two of them are just an agreement of some kind. First one is an agreement on how we structure our exam cards on scanner console, in our case it's Siemens, which defines naming and placement of datasets in a hierarchy later on when we do conversion. Second one is ReproIn specification, which looks like bits, but it's not exactly bits, of protocol names. It's pretty much how do you name your sequences in the scanner. And that defines naming and placement of files within those bits datasets, which are composed into hierarchy. And then there is a third piece, which is actually finally fun part of coding, which implements our ReproIn conversion through HeuDiConv. So there is three independent parts. You, as a user, could pretty much use either of those. Well, third one doesn't make sense without one and two, but two could be used without one. So if you don't have such nice hierarchical organization on the console, you could still use ReproIn just to do conversion. And let me talk a little bit more about this hierarchy. All the materials for ReproIn and HeuDiConv are available online, primarily on sources are available through GitHub, but also if you go to ReproIn GitHub page, README there provides sufficient description and pointers so you could start using ReproIn with HeuDiConv. And one of those documentation pieces is a walkthrough which guides through our setup on Siemens console at our center. So let me give you just a few examples which are related to this hierarchical organization of data sets. When you go into .cockpit or Explorer of Siemens Scanner, you'll see that there is regions, and then let's say this region, I believe, is Siemens, and they have already a collection of sub-regions, I would say, and within those they have exam cards. So we decided to abuse this structure for our good cause, and decided to use first level of the structure of region to create folders per each investigator. So every investigator in our center, they have their own folder of a kind where they collect their exam cards. And then next level, which is optional, you don't have to use it. So you could just have an investigator there. But typically, it's not the PI who runs the study. It's some postdoc or student, and you could annotate there. You will use the same investigator, but then use underscore and put postdoc or student name there who actually runs the study. And then within there, you'll create your collection of exam cards with populated with the protocols for these sequences which you want to run. Why is that necessary, or why we had to do it? It is because this is the only way we found, so we could avoid any manual entry, so people don't have to enter investigator or experimenter within the initial exam card window. But when they select a specific study, like in this shown here, it populates study description, which is later on transmitted within DICOM files. So that fulfills our kind of desire to minimize manual entry and have sufficient amount of information shipped through the DICOM so we could organize our data sets in a logical hierarchy. And if you go to GitHub page through this walkthrough, it details it it guides you through each step, so you could start using it on your Siemens Prisma. So hierarchy is, as I mentioned, is optional feature, right? If you, in your case, you don't have such nice hierarchy, when later on you will run HeuDiConv, just specify locator empty string, and as such, it will not use any hierarchy, or you could specify the same underscore separated locator, I believe, and it will place it under in a specified hierarchy. So this is an example how it looks at our center for a specific case. In this case, we have top level directory bits, which contains, again, subdirectories per each investigator. In this case, as I mentioned, I stole slides from this particular slide from Matteo's talk. And this is Matteo, right? So he was running a number of studies while he was part of Gabini's lab. Now Matteo is somewhere in California, and unfortunately, we don't see more studies appearing here. But for these studies, all the studies which he ran within Gabini's lab, they're nicely organized and placed in the specific location on the server. So later on, PI Gabini could go and find any data set which Matteo or Vasiki or any other student ran in her lab. The same goes for any other PI. So this is overview what you get by simply investing maybe additional 5 minutes into organizing your exam cards straight on the console in a logical hierarchy. And the second part of the recipe I've mentioned was specification. And again, it's pointed to from the same GitHub page on ReproIn repository. And when you click on that heuristic link, you will be redirected to HeuDiConv, and actually to specific file within HeuDiConv, which is shipped under heuristics reproin.py, where in the comments of that file, that's where the specification for ReproIn lives. So ReproIn is just this third piece code is just a part of HeuDiConv distribution. So any user who installs HeuDiConv automatically gets access to ReproIn heuristic and could convert any data set which was acquired following such a heuristic. And heuristic at large, specification at large, follows bit convention. Like you could see there is a task, there is a acquisition label, there is a run. So you could put pretty much any bits specific key value pair in your sequence name in the scanner. And the only difference that you didn't need preamble about subject or session, well, although session is available, so you could specify specifically which session. But also we will ignore and strip any prefix which specific scanner might add, like work in progress sequences or prefix provided, I believe, by, I don't remember who provides it, or you could add your own prefix if you like, separated with a column. So here's an example, again, from Matteo. So under Gabini's lab, there is Gabini Matteo folder, and then all those studies. And let's say in this study for fem-face angles, he had to run 2 sessions. That's why he created 2 exam cards in which he populated specific sequences he wants. And you could see that they all look similar to bits, besides that they have prefix which abbreviates for the modality which that sequence is for, either for anatomy or field map or functional. And one of the sequences, Scout, has annotation of session. So that's a tricky part we had to work out because, as I showed before, when you select this exam card, it populates study description, but it doesn't use this next level. It just uses previous levels and doesn't use this level or specifically a name of this exam. So we had to add session annotation within a name of single, or it could be identically present in all of them sequence. And what happens after we scan, our scanner immediately sends DICOMs whenever any sequence is finished, and they're cataloged by PEX server into a directory for that accession with names of the protocols in the directory name. But it's not even necessary in the sense that ReproIn heuristic doesn't care about what is your directory name. It cares only about information which will come from the DICOM files which you'll provide. So one only piece which I didn't talk about and is missing, where do we specify the additional information such as subject ID? So, and that's where patient registration card comes in, where date of birth and patient ID and weight are specified and the gender, and all that information is actually does get populated in the bits DICOM files, so we could later extract it and place it into bits datasets. Again, more information available from that walkthrough. And as a result, after running HeuDiConv with ReproIn heuristic, which underneath uses DCM to an AX, we could get our DICOMs nicely laid out as bits dataset files automatically. So overall, how to use HeuDiConv with ReproIn heuristic is as simple as running HeuDiConv, specifying that I'll use ReproIn heuristic, that it will be bits dataset, and in this case, we also annotate that it should be under data-led control, which I'll mention a little bit later, and where to place the resulting dataset with this hierarchy I've mentioned underneath, if there is a locator information within DICOMs. And then you point to the files which it needs to process. And these files could be just a directory with multiple accessions. So it doesn't have to be a single accession or a single session. You could point it to, let's say, all DICOMs you have for the study, and then HeuDiConv will sort them into accessions according to the metadata contained in DICOMs, and will organize them nicely into different studies and within this hierarchy which you have. There are examples. So again, if you go to ReproIn GitHub repository, it will point you to the data which we share publicly, which was collected on Phantom, so we could share those DICOMs. And there is quite good collection of those just to try HeuDiConv with ReproIn and some DICOM data. So what ReproIn and HeuDiConv feature overall? That all those sidecar JSON files get automatically produced by DCM29X. Actually, HeuDiConv also enhances them with additional metadata, which it extracts from DICOMs, unless you specify option for minimum metadata information. Then ReproIn heuristic stores original DICOMs under source data following exactly the same name convention as original files and bits, just storing them within tarballs. So later on, if you want to look into DICOMs which are associated with specific Nifty file in your bits dataset, you have clear way, because it's exactly the same path, only under source data, to get to those DICOMs. But also you could use those sort data to, let's say, reconvert all the data later on if there is necessity. Then HeuDiConv also creates placeholders for bit sidecar files and some top-level files such as README, dataset description, and prepares you to fill out events files. And then all this data-led component comes in. So in our case, we really like version control systems, and DataLab is our project which allows us to version control everything, not just code or text. It allows to control data files or complete containers, images, so we could have all necessary information version controlled. Also, as part of a ReproIn project, we implemented support for faking Git commit dates. And that was implemented so later on this bits datasets which produced with HeuDiConv don't leak any information about which might be, let's say, if you process data right when it gets from the scanner, the date of visit of the patient might leak this way through the Git commits dates. So there is a feature now in DataLab where you could create a data set where every operation will commit with a fake date starting from April 7th of 2005 with 1 second increments. That could be a challenge to you to figure out why specifically that date and time, which is an interesting question on its own, I would say. And also another feature of DataLab is modularity of the data sets. So we call it data-led sub-data set. So a data set could contain sub-data sets within it, which I will demonstrate now as a bigger overarching goal, what we want to achieve to help with reproducibility, but also just gaining information to all components of a study. Let's say in this case, every squarish block here is data-led data set. And we could see that through this sub-data set organization, where we could say that, okay, bits data set is derived from raw data. So it could contain raw data sub-data set, which can be optionally installed or uninstalled. And then based on this bits data, we did pre-processing using fMRI prep. So fMRI prep dataset in turn contains reference to bits dataset. So later on, you could install either the entirety of fMRI prep dataset without bits or with the original bits dataset in it. And it goes kind of all the way up. So you could create a dataset for your analysis A or B, and those will point to specific fMRI prep dataset, and it propagates all the way to the paper. So at the end, you could have your publication repository or a dataset, because dataset is just Git Annex repository. It could have capability to track down all the way to the raw data and have clear versioning information. But as I've mentioned, also containers themselves could be a part of that data set. So you have access not only to the code, to the data, but you have access to the entire containers which were used to do actual analysis. And such organization is part of our HOWTO principles. I would advise you to visit our ReproIn containers repository, which has all the additional references to HOWTO and DataLab. So having this hierarchy, so everything, HOWTO principles could be fulfilled using DataLab. So I've mentioned datalab many times, but if you want to discover more, just go to datalab.org. And if you want to familiarize yourself much better, then go to user handbook. So if you click on this link, you'll be redirected to handbook.datalab.org, which will guide you through the basic principles of datalab and also give you instructions on how to get it installed and get started. So let me get back to our example here where we were converting specific study from Mattel. As I've demonstrated before, all data set get into this hierarchy where for specific study, all the components of the path pretty much come from DICOM. But also additional feature of having data under data lake control that underneath there is just all the data is contained within Git repository. So you could use regular Git commands such as git log, git short log to discover what was done to the dataset, by whom it was done. In this case, git short log demonstrates that our dbig bits team was doing the initial conversion and maybe added some additional content. And then Mateo was the one who curated the dataset, right? And all these actions are described within your git log. And sometimes some specific examples, some specific changes recorded within Git history could be eye-opening later on. What happened to my subject 14, right? So apparently, there was bad conversion, then it was reconverted later on. And you could see that additional curation might be necessary whenever there are duplicate runs. So you have ability to get this data set from the scanner, do curation, and then use facilities of datalab, which pretty much facilities of Git and Gitanix, to introduce new data from the scanner. So you could start analyzing your data set while not losing track or overwriting your changes with old files from the scanner, or even just merging two histories together, which is what Git is great for. So overall, HeuDiConv ReproIn HOWTO summary for you, how to adopt it. And there were not that many commands, but the main principle is to adopt our naming convention at the scanner. And you could use it with or without hierarchy aspect of the convention. Then you could use HeuDiConv with ReproIn heuristic file for your data sets, DICOMs, which were named, had sequences named according to our convention, but also it's possible to use it even for already collected DICOMs. We have examples like that, and we just need to provide how to in the same GitHub README. So maybe within a week we'll do that. There are Docker and Singularity images available, and also ReproIn container is available within ReproIn containers. So you could use this data-led dataset with many containers inside for all the beads apps and also additional ReproIn products. So you could have a nice single point of entry and also really nicely version controlled containers. Additional advantage is that everything is version controlled and you have a guarantee that those specific versions of the containers will always be available to you either from our website or from Singularity Hub. So part of the ReproIn project will be to guarantee availability of these containers even whenever, let's say, hub of the Docker disappears or something happens to Singularity Hub. And additional how-to is not to stay silent, but if you have ideas or recommendations or bug reports, contribute to our GitHub repositories. So what else we have planned is to make these customizations, let's say when you've made a mistake in how you named your protocols in the scanner. So we want to make it easier to provide these customizations per each study. Also extend it to other modalities. So far, I don't know, use of ReproIn convention, let's say, for collecting EEG data, and nobody looked into how we could collect enough of metadata within EEG files. So such layout could be automated as well. Then another project, Repro Events, I believe we call it, would be automatic generation of those events files with events from the stimuli and also trigger pulses from the scanner. So you don't need to worry about generating those if your experiment is, let's say, specific task, not just resting state. But also we want to automate collection of audio-video stimuli as it was presented to the subjects and others physiological data. If you want to join, we have positions opened, so please get in touch. And additional related references is that ReproIn or HeuDiConv is not the only solution for conversion of DICOMs into bits. Bits website lists additional toolkits. And one of those other ones is Data-led HERNI from our collaborating project number 5, which provides web UI for flexible automated conversion of any raw data, not only DICOMs, into target layout. For DICOMs, they use underneath HeuDiConv, and for organization of data, they also use Data-led, and it should be possible to use HERNI with ReproIn convention. Also, if your center has flywheel management system, then there is flywheel gear for HeuDiConv, and because ReproIn heuristic shipped within HeuDiConv, you could use this flywheel gear for your data through flywheel system. Thank you for your attention. I would be glad to answer any questions, and if you have any issues or recommendations, please file them on GitHub.
Thank you, Yark. That was, as always, very informative and clear. For any of you out there in listening land, if you're daring to try to challenge your ability to unmute yourself on Zoom and have a question you want to ask, now is the great opportunity to do that.
Okay, I have one. ReproNim is for neuroimaging. And what's the in? Is it just in?
Input. Reproducible input. Yeah.
Thank you. That was an easy one.
So have you invented Repro out yet, Yark?
Repro. Data-led. We should rename it.
Or the Repro Pub. It could be AKA.
That's what I was going to say. It's the Repro Pub.
Yeah. Okay. Thank you. We have some new terms to use. Hearing no additional fearless people who ask questions, I will thank everyone. There's been ample information here about how to contact Yarek or to file issues. Again, there is lots of support for issues on the websites listed. These slides will be available. I will tweet out and will email out the repository where all these slides live, and the video recording of this will also be available as well. Thank you all for joining. Again, next webinar will be January 3rd, again at 2 p.m. Eastern, and I am even more convinced that it will be the test Kraken suite of tool testing resources that will be presented by Dorota and Satra and the MIT team. So again, thank you, stay tuned, participate, and have a great day out there.